Hello, everyone, and welcome to the Xavier AI Experts Network. Today, our agenda will be going through 5 minutes of introductions, our mission, and the co-leads. We'll walk through the presentation and save discussion and future topic selection for the last 25 minutes. Our purpose is really to establish a collaborative forum where AI experts across academia, government, and industry can come together in the purpose to accelerate the development of AI solutions for the benefit of our global healthcare community. The intent is really to encourage a free flow discussion and idea exchange. We are not aimed at creating white papers or providing recommendations at this time. And then you can also find additional information about the network in the link provided here below. My name is Simon Perkowski, so I'm one of the co-leads here alongside with Arvind as well as Kirthi. Kirthi wasn't able to join us today, but Arvind will be helping facilitate the discussion throughout the talk. All right, and now it's with great pleasure that I introduce our featured speaker, Toni Manzano. Toni is a co-founder and chief scientific officer at Aizon, a cloud company that provides a big data and AI SaaS platform to the biotech and pharma industry. Since 1996, he has led software projects for international pharmaceutical companies, covering the entire production process and supply chain, everything from R&D to clinical trials, quality assurance, to warehousing and logistics. He worked as a researcher at the University of Barcelona as a physicist, and he teaches big data and artificial intelligence subjects related to the pharma industry at the university. He has written numerous articles in the pharma field and holds a dozen international patents related to the encryption, transmission, storage, and processing of large volumes of data for regulated environments in the cloud. And with that, Toni, please take it away.
Thank you so much, Simon, and thank you so much for all the audience for attending this meeting. Please chime in during the presentation because I like to answer and to share thoughts and comments and questions during the presentation because it's like a dialogue. It's not just a very boring presentation. Well, first of all, let me start with 3 definitions. I know that this is very hard to start, but usually when I'm teaching at the university, we cannot avoid that. So we need definitions, and afterward we can go to the practical part. So let's start with this definition. Probably for you, if you're not coming from the pharma industry, this is very obvious. So manufacturing science is a concept that was defined in 2003, a lot of years ago. And they defined manufacturing science as the combination of many values, many variables coming from efficiency, coming from process efficacy, coming from product quality, coming from human interactions. So they define manufacturing science as the combination of all the involved factors in the manufacturing processes in order to know and to understand, to get knowledge from the reality behind these manufacturing processes. If you are coming from the pharma industry, this is a little bit shocked. Why? Because usually in the pharma industry, we have in one hand process, in the other hand, we have quality. And when we are focusing our attention in the process, we have to ensure the efficiency of our process. We have to maintain our overall efficient equipment, the OEE, with a very high level in order to be efficient. But on the other hand, for pharma, we have to produce drug with quality enough. So usually, traditionally, in the pharma, both concepts were not always going together. This is important because from 2003, this concept was put in place. The second definition that I like a lot is PAT, PAT, Process Analytical Technology. This concept was introduced by the ICH, the Immunization Organization, in the Q8 in 2009. They aimed to control a process, getting all the knowledge on this process, being able to act if it is needed in order to fix any kind of potential issue that could arise during the process. So again, if you are coming from the pharma industry, you will know that it's very difficult to act without any kind of rules in the process in order to rearrange, to solve some potential issue. Well, the thing is that in 2009, they presented the PAT concept as the way to control, in a regulated way, some processes that could be managed during the ongoing execution. And they define that the only way to do that is having a multivariate knowledge, a complex understanding of the variability of the process in order to be able to act. So this is a very old concept, 2009, defending or promoting precisely the AI abilities. And finally, this is the oldest definition that I found regarding control systems by AI. So in a lot of years ago, control systems were defined by two different attributes, controllability and observability. And precisely if you look for these sentences in the ICHQ8, you will see a lot of equivalence sentence pointing out to this reality. Everything that you want to control must be observed. So usually when you work in, not only in pharma, in other areas, a process is like a black box. It's like it's a process that has an input, has an output. We have to wait for the output, but we really don't know what's happening inside. So AI is a very good tool for controllability and observability because we should be able to understand not only the input and the output, also what's happening inside the process. And that's all. So this is the three definitions that I wanted to introduce you. If you don't have any questions, let's go to the practical part. The first one. So now I would like to introduce you two real use cases of AI applications in pharma. The first one is this one, continued process verification, CPV of the future. CPV maybe is an utopia. So a lot of many pharma industries, they're looking for implementing continued process verification in order to ensure that every single batch that they produce is a good batch, is an optimal batch. The problem is that to do that, you have to act on real time with the process, with the ongoing process. And sometimes, or very usually, the process is always controlled by variable, looking at the specifications variable by variable. But the reality is different. The reality is a complex reality where all variables are interacting continuously between them. Let me introduce you this nice, amazing project, CPV of the future. This project is being led by the PDA, Parenteral Drug Association, and is funded by the Product Quality Research Institute, where some members of the FDA, they are in the steering committee. Well, the thing is that this project led, as you will see, for several pharma companies, top pharma companies, Sanofi, Lilly. We are working also with two universities, Maryland and also the Barcelona one, where I'm teaching, and also AstraZeneca in this use case, and also the Xavier University in the AI in operations team. Here, Mario is representing this team. Which is the goal? The goal of this project is to demonstrate that AI is the tool, the multivariable tool to control the process when you have enough knowledge in order to control the process, considering that the process cannot be managed variable by variable. So all the variables are interrelating between them, and you should control all of them at the same time if you want to control the process. This is the main aiming. Just to let you know, we have already finished the paper explaining the first phase of this project, and now is on the review, in the peer review. Let me start explaining which is the basics of this project. This is the workflow or the metabolic flux of the PK pastoris. The PK pastoris is a yeast that is very well known in biotech and also in biopharma, because this yeast, this fungus, is able to produce lipase proteins that we use for drugs. Well, you can see here that the metabolic workflow is so complex. There are many, many paths and different paths. But the thing is that there were some discovery around a specific path where you can reduce the oxygen during the metabolic workflow. And it seems that the fungus is able to produce more and more lipase in this case. So you can accelerate the process. It's like with the same amount of raw material, you can produce the double of the beers. Let me say this is the example. So we can multiply per 2 the amount of final product, just decreasing the level of oxygen. Well, and this is what's the focus: how to control, how to determine which is the optimal moment, how we can define the optimal amount of glucose, the optimal amount of ethanol that we can deliver to the picky pastores in order to get this double amount of output. From a biotech perspective, usually we divide the biotech processes in upstream and downstream. Upstream is when we are harvesting, we are creating this final output. And once you have this bulk material, you need to filtrate, you need to purify this material. This is downstream. In this case, just to put the focus in where we are in upstream process during the fermentation process. And we use experimental batches. We use real use cases in order to demonstrate this assumption that AI could be a very good tool. How to start? The regulation says that if you want to get knowledge, there are many good practices to get this knowledge. One of them is defining a right DOE, design of experiments. So you can design all the happy paths, corner cases, usual cases, in order to describe how your experimentation in real life would work in order to identify potential anomalies or whatever. Well, we try to do that using the subject matter expert knowledge. And we discover that, well, the things that they are using usually is that they are considering linear approaches for all the variables that were involved. And we determined that with 10 experiments, 5 under hypoxic conditions, so reducing the level of oxygen, and 5 experiments providing the normal rates of oxygen would be enough in order to cover all the experiments. And we preserved the 11th experiment in order to validate all the AI models that we created. This is the design of experiments. Well, you can think about, well, this is not too much data. This is the first thing that we can see from an AI perspective. 10 batches is not too much. But this is how the university works. We don't have too many resources. And this is how it's costly for us. What we did in order to get more data. This is a strategy that we usually use. And could you see here normoxic and hypoxic conditions? We had a lot of CQAs, control quality attributes at the end. And in order to reduce or to multiply the data, we split each process in 4 phases: batch, adaptation, early fed batch, and later fed batch. For every single process, we split the process in 4 phases. And even more, we had more or less around 20 CQAs, 20, let me say, target variables, 20 CQAs. How we can aggregate that in just one? Because we don't have many data. Well, we asked to the subject matter expert to categorize each phase of each process in one of these 3 areas: good, average, or bad. With that, we aggregate the 20 CQAs, the 20 target variables, in just one variable that was classified in 3 values, 3 categories: good, average, and bad. And this is the variables that we were working with. well, the name is not very relevant for this talk, but just to let you know that in this carbon emission rate, oxygen uptake rate, and respiratory coefficient, we are also including some of the independent variables in the calculation, but sometimes not. And this is the analytical expressions to describe the behavior of the metabolic flow in the PKF stories. So just looking at that, we easily identify that this is not linear. Please, we cannot think in linear approaches if we are looking at that. But I don't know why, traditionally, we are using always correlations or even more. For every single process, you can see here, this is the name of each batch, each process 2, 3, 4, 5. For each process, the subject matter experts try to explain the relationship between the variables using, for example, PCA. And this is the PCA, the principal component analysis, and the relationship between the variables, always the same variables, remember these ones, how they were related between them for every single process. And of course, the relationship were not consistent for every process. Why? The reason is that the reality is always containing variability, not only explained by the variables that they are defining the metabolic flow in a very lab and small conditions. The reality is very different. The ambience, temperature, humidity in the lab, everything is changing. The operator is changing. So depending in how you are establishing the setup parameters, everything is changing. So we cannot consider that our systems are linear, because we demonstrated that using a PCA, the relationship between the variables were not the same batch by batch. Well, what we did, we tried to identify if at least putting all the variables without to know anything or without to consider, let me say, the subject matter expert opinions. I think that we have something in the chat. Oh, no, sorry. We consider that putting all the variables together without this previous knowledge, we should be able to identify if at least these variables are explaining or not the problem. Well, we did that, and we used pattern recognition for every single variable across all the batches that we had, in order to identify the pattern recognition using the cosinus similarity was able to identify good phases, average phases, or bad phases. And the result was that, yes, the algorithm based on this pattern recognition for the cosinus similarity was able to identify this difference between these categories, good, average, and bad. That means that at least the variables that we are considering, they are explaining well the problem, but beware. So we have not to consider linear dependencies. They explain the problem, but probably they are not linear. This is very important. What we did? Well, the first thing that we did was to control potential anomalies or deviations that could arise during the process in an unattended way. So imagine that you have an operator 24/7 looking at the process, continuously identifying if there is something that could be out of specification. And here, let me say an important topic. When you are in pharma or in other areas, in semiconductors or whatever, and you are looking at variable by variable, and always these variables are under specifications, you would be happy because all of them, they are under control. But what happens when, although all variables are under specifications, maybe some of them, they are going out close to some of the limits, upper or low limits. The process is under control because all the values are in their specifications. But for some reasons, the process could be going out because the trending or the combination of variables are moving to a non-expected condition. Well, this is what we did. We applied here isolation forest. The isolation forest algorithm is very good to detect anomalies with just a few data. And we applied this outlier detection, this anomaly detection using the isolation forest. And we classified the anomalies based on extreme severity, medium, and low severity. Well, we applied that in the historical data, and this is the, we detected many low severity issues, and we confirmed them with the subject matter expert. Here, the good point is that we created this isolation forest model hour per hour. So we couldn't do that for all the process because we needed to create a model specifically for some time windows in order to get the specific behavior. Remember, we're talking about some microorganism, the Pichia pastoris, is alive and is reacting with everything in the lab. Could you see here, this is the 5 liters bioreactor, the temperature outside, the hands of the operator, everything is interacting with that. So we cannot conclude that with just linear or with a model to cover all the process is enough. We needed a lot of models determining in which subphase we were in each operation, let me say, in each phase. This is the first thing that we did. The second thing that we did was, what about if we try to recommend some critical process parameter, some ongoing parameter that is manually controlled by operators in order to ensure that the final goal, the outcome, the LIPAs that we're producing is optimal always. Although this parameter, this control is nowadays in big pharmas made automatically, this control is usually made using classical approaches. So we have two specifications, two limit of specifications for high and low, and two more for high, high, and low, low. But what about if we try to provide the optimal value always under the control? And that is what we did. And we create using a simple, very random forest. Believe me, a random forest was enough in order to provide the predicted agitation speed. This is the black one, is the agitation speed that we propose to the operator to apply. And the blue one was the agitation speed provided by the formula that this process had, always under control. The black one that predicted was also under control, but was, depending on in which phase we were under the recipe, and at the end of the process, we were up to the recipe. Well, the thing was that at the end, we got very good results. We checked that with the subject matter experts at the university. We checked that in the Pichia pastoris event in California this year, and it was really successful results, demonstrating that AI can be very good monitoring the process, alerting if there is any kind of deviations or anomaly, and predicting or recommending the optimal parameter in order to get the maximum yield at the end. But even more, we were not just happy with that. We thought, what about if we try to create more data? Remember, we were just working with a very small number of batches. What about if we try to create synthetic batches? And this is what we did. So in this case, we created a synthetic data generation process based on the experimental data that we got. Here, we didn't make any kind of assumptions. We work in a very blind way in order to don't include any bias. And we divide the variables in two groups: the dependent variables, the ones that they are defining very well the quality of the product, the CQAs, and independent variables, the 7 control parameters like temperature, pH, and ethanol, and whatever, in order to define these synthetic batches. Well, we create these machine learning models based on that, and we got very high accuracy models. which was the main hypothesis. The hypothesis was, what about if we consider that the product quality is a function of the time? Probably everyone knows that, but the quality of the final product depends on all the process, all the values that we have achieved. It's not just a question of the final result. So we have to ensure that the product quality is always guaranteed from the beginning, since the beginning till the end, not just at the end. So this was the first assumption. And considering this function of the time, the CQA, the quality control attributes, is this function at the end. It's just the measure that we are getting from the this carbon rate and the rational, the respiratory coefficient and so on at the end. But they are really a function of the time. Considering that, we created these machine learning algorithms, and we try to create two kinds of algorithms. One of them we're using, for example, for all those CPPs, all those parameters that would be described by a function like the microarray. We used functions, simulating this data using functions. In this case, for example, we use an exponential function. But for all those parameters that cannot be described by a function because it's very, let me say, ad hoc or random variables like the ethanol, we create a custom Monte Carlo algorithm in order to produce this data with some parameters, with some parameters that we can configure. Well, the thing is that we try to work with different combination of these parameters, and we got several that were so rigid, several that were producing very noisy values, and we discovered, just working with different games, let me say, we discovered that this combination of parameters were very good data, synthetic data for all the variables that were involved in the batch. Producing a lot of batches, here we can see 40 synthetic batches versus the 4 experimented batches, we saw that all the variables were really good, and they were inside the extremes of the maximum or the better and the worst batch. And now in this, once we finish that, nowadays we have a machine learning with a complex system that they are able to identify or to create batches for every single variable, what we can play with. And in the phase 1, that this is this experiment that I have explained to you, where we are writing now the paper that is under peer review. In the second phase that we are working now, we are using these synthetic batches generated in order to create more experiments in the design of experiments. This is the goal.
Toni, sorry to
please.
interrupt.
Please.
This is Arvind. Quick question, I think came up in the chat. Betsy had a question. Betsy, would you like to ask, or would you like for me to read it out?
Yes, the question from Betsy says, maybe you will cover this later. I see you talking about process monitoring. What about control against CPPs and automatic adjustment at critical points in the process? Yes, you're right. Betsy, this is the second phase of the project. Just to answer you, now in the second phase where the Xavier in operations team is working, we are trying to control automatically using an adaptive phase, using an interactive phase with the bioreactor, providing the right values for all the parameters at the same time in order to get the optimal CQAs at the end. In this experiment, we wanted just to demonstrate that something is able to do with AI, something we can improve with AI. Now we are gonna go beyond that. We're gonna use all the AI algorithms that we have in order to provide the optimal values for every single CPP that we have. Second phase, we're working now in that. I don't know if that answers your question, Betsy.
Yes,
well,
yes, thank you.
Just,
yeah, thanks.
A quick question about following on that, Toni, which is, oh, I think it's already good questions come up.
Yes, we have another one. Since
yeah,
you are building, yes?
Go ahead.
Since you are building models on an hourly basis, how are the predictions during transition from one hour to another? does it become abruptly discontinuous, or do you do something to soften it? Well, it's curious because we was very worried precisely about this quantum process, let me say, 1 hour, 1 hour. So you're completely right. But we were really surprised when we saw that was the models themselves, they were really smooth because we provide some inputs at the end of each hour to the model coming from the next hour. And the response was really, really good. So it seems that the models themselves, the isolation forest, was responding very well between transitions because at the end of each phase was very well related with the beginning of the next one. So it was not needed to do any kind of smooth process.
Thank you.
You're welcome. Okay, so I hope that this is not too boring. The next one is more biotech again, but this is more related with direct drug. In this case, we are talking about a real top 5 plasma pharmaceutical company around the world. Provided you already know this problem, when you work with real use cases, you cannot share the name of the companies. And for this reason, I'm very proud to share with you the data coming from the PDA and PQRA, the CPV of the future project, because it's an open and scientific open project that we can share data, we can share results, and everything is shared. So I'm very happy with that. But in this case, well, we can share something, but not everything. Let me share with you some of the main takeaways of this use case. This is a plasma company. So this company is doing drugs, is manufacturing drugs for humans based on donations, on plasma donation. So you cannot imagine how much variety there are in the raw material, because at the end, this company is getting blood from everywhere around the world. So donors are bringing their raw material, and the process in order to create these drugs is so complex because there are many sequences, many phases. But at least just to make it that very simple, they are using a lot of centrifuge, a lot of filters, a lot of centrifuge. So they are separating all the phases on the blood in order to get different drugs. Okay, so at the end, this is the main principle: centrifuging a lot, filtering a lot, and you get in every single step, you can have different drugs. This is more or less. Well, the problem with this company was that consistently during the last 4 years, they were experimenting a decrease, a systematical decrease on the yield of the final drug. And well, you can imagine that the problem is coming from the raw material, of course, no? Because if they are doing the same, they have all the process already validated, everything is under control, and they are systematically having a decrease on the yield, the only source of variance is the raw material, of course. But how we can understand how to control that, and even more, how we can improve the process and increase the yield if it is possible, interacting with the manufacturing processes. Well, they had, this company has a very good team of statisticians, but the problem is that they were not aware about the added value of AI working with multivariability, complexity, and variance. So they were using, for example, PCAs in order to describe the relationship between the variables. They were trying to describe statistically how the raw material attributes were affecting to the yield at the end. But the problem was that there are many processes in the middle that they are provoking some perturbances in the process. Well, at the end, I would like to advance the end of the movie. We were able to increase the yield, and we are very happy just applying AI. This is very good results. And let me explain you what we did. Well, the problem was that in 4 consecutive years, because that happens 4 years ago, they were starting to detect this systematical decrease of the yield. Well, which was the root cause? We know that there is a large dispersion on all the variables. And the first approach that we did with them was to try to make a correlation between the yield and the quality control quality attributes year by year. The problem was that we were trying to do that from a statistical perspective, doing univariate relationship over the time with the yield. After that, we didn't got any kind of good result, and we tried to apply PLS. What happens? Well, as you can imagine, we didn't find anything that could explain the problem. Why? Because PLS is describing a linear approach between the variables. And usually when you are working with complex things and with a continuous variability, the linear approach is not the very good one. What we did? Well, we tried to create clusters of batches depending on the origin of the raw material of the donations, and automatically it appears 4 clear clusters describing the yield for every single cluster. So remember that in this case, we work very closely with the subject matter experts, trying to figure out why these clusters were appearing in the scene. Just a key k-means, k-NNs, very simple AI models, but the 4 clusters appear very quickly. And afterwards, we created and stratified correlations by clusters. This is important. Well, the problem that we had during the process was the following one. There were not contextualization of data for all the offline data. So we were using a lot of data coming from the historical information, but there were not any kind of context. For example, what happens if the raw material is coming in summer? What happens if it's coming in spring? What happens if, which is the external operation where the drug is delivering the material, although it's in under cool conditions, there are very short time of windows where there is a difference of temperature. What about the data that we are using? Because many of data that the customer provide to us was in Excel spreadsheets. How we can rely on that? Okay. Well, we use this cluster 3. We use this correlation. Let me see correlation based on these 3 clustering, and it seems that the process were very clear, creating this kind of groups using just a very simple k-means based on the yield. Well, with that, we were able to identify these 3 groups. You can see here these 3 groups, where the darker colors means that there is some a better yield. In this case, I have to mention that we were working with anonymized columns. We didn't know anything about the variables. So F96, F84 were variables that were completely anonymized for us. And we worked just with pure data. Well, what we found in these very good batches and in these wrong batches? Well, we found that the shorter the process is, the higher the yield is. So what means shorter process? Is because people is going fast? Is because there is less issues? Is because there are less downtimes? Which is the meaning of that? But well, it's something that was thanks to the contextualization that we add in the data. So remember, I think that when we have, or when you are working with data, it's so important to create context on this data. The duration of each phase was not included in the initial information that we got. We added this column and other columns, like for example, Aizon, and we found very nice relationship between them. Well, just working with duration, we found the very good relationship between the yield and the time that people was spending in order to produce the yield. Remember, always working with anonymized data. And it seems that the stability of the process, so isn't, let me say another way. When you are working with this kind of plasma, this plasma, depending on which season you are, is bringing some intermediate times where the plasma has not been completely right in the cool conditions. For example, when you are downloading from the track to the silo, although the silo is under control, the track is under control, there is a very small time where you can have this exchange of temperature depending on the season. Well, the thing is that depending on how much time the process is, the plasma is degrading less. And we found that the duration of the process, and specifically in some specific phases at the beginning of the centrifuge processes, were affecting a lot to the final process. Let me share with you that this kind of full process with 6 centrifuge operations, usually they require more or less 2 or 3 days. So how you can relate from a statistical perspective that the initial phases provoke some perturbance at the end of the process? It's very difficult if you don't use a multivariable tool. Well, this is the result. We use a very simple random forest to create these predictive models. Here you have the columns with the anonymized variables, and 2 variables, specifically the D49 attribute, was the most important feature explaining this yield at the end of the process. This D49 attribute is very related with the time in the previous phases of the manufacturing process for this plasma. And that's all. It's just to share with you 2 real use cases in pharma where AI is bringing value. And when we consider that the system is not linear, when we try to understand that the system is always variant, is not constant, and the complexity cannot be understood from a monovariable perspective, I think that using just very simple AI algorithms, we can get a lot of benefit. This is just the summary. I'm happy to share thoughts, reflections with all of you. Thank you so much.
Thank you, Toni. Wonderful talk. Very great case studies and learning opportunities. I think this is now a good time to bring up any questions for Toni and learn from his experience. Please feel free to either put them in the chat or you can unmute yourself and ask questions. So while we are waiting, perhaps if you don't mind, Toni, I'd like to start things off. You talked about data-driven function fitting. I found that picture that you drew for us between the exponential function and the data-driven Monte Carlo simulations. Is there a package that you would recommend for this kind of function fitting that you think is especially helpful?
Well, we use a lot of Skylab libraries and also TensorFlow, but we were using just the exponential approach coming from the math library in Python for this exponential one. And for the Monte Carlo, we use also a specific library that we have in TensorFlow for Monte Carlo.
I see. Got it. Yeah. Thank you so much.
You're welcome.
Our follow-up question was, as you fit these functions and they're part of these generative models, I think you did speak to it briefly when you talked about variance and those kinds of things. What is the expectation on the calibration and the uncertainty emerging from these predictive models? What's a good set of practices to adopt while reporting the credibility of the models that you are building?
Good point. So for the second phase of the CPV, where we are working now, we are going to use synthetic batches as part of the design of experiments, because usually the design of experiments sometimes is so difficult to create the experiments in the corner cases. What we are going to do? Well, with these synthetic batches, we're going to fit the formulas, the analytical expressions that we have in order to explain the metabolic flux of the PK pastoris, and we're going to use the synthetic batches in order to fit these analytical expressions in order to know if the analytical expressions are providing good results or not. For example, imagine that you have an analytical expression that is describing or is providing you the respiratory coefficient. Well, putting all the variables that we are generating synthetically in the expression, if the respiratory coefficient is good, that means that the synthetic batch could be good. This is one of the verifications that we are doing. The second one is that for every single synthetic batch that we are going to use in the design of experiments, we will double check with the subject matter experts in order to know if they make sense or not.
I don't know if that sense. That makes a lot of sense. So on that note, I mean, you mentioned the notion of data paucity, and it was very clever how you were able to leverage even what would have been considered a small amount of data for in a very clever way. And I think a couple of questions that were already asked during your talk made sense in that regard. I wonder, in terms of trying to merge the knowledge that we have cultivated in this domain of process assessment, there's a lot of physics already at play. And I understand that you have a background in physics, so hopefully this will make some sense as I'm saying it, which is the physics of the problem already gives you a limited hypothesis space to work with. Yet we have all this data that is nonlinear and is shifting in time, etc. What's the best way to merge what's happening with AI, the limited amount of data, and the physics of the problem, so that we can leverage even small amounts of data in clever ways?
Well, the thing is that the reality is that although the physics are explaining very well the context from a lab perspective, so the lab perspective, as probably you know, this is not real. This is very controlled, without friction, without complex interactions. So physics are explaining well the optimal conditions. And we can have some batches in production that, well, could work under optimal conditions. But the reality is not this one. Even in physics, I'm in physics, and all my career was always worried about how to make approaches, how to ignore, how to make obvious or ignore the friction, how to don't take care about whatever problem we have that is not in the formulas. Why? Because the reality is always more complex. So I think that physics works very well when the process is in optimal, under normal conditions. And we use physics or analytical expressions in order to double check what's happening with the happy path in AI. But the problem is what happens in normal conditions. Normal conditions means you have problems for corner cases, non-happy paths, when you have unexpected or unexpected reactions. So this is how the relationship is. Sometimes you have physics that can be checked or can be double checked with AI, but in many cases it's not possible.
Makes a lot of sense. Thank you, Toni. I've been asking too many questions. I wanted to open it up.
No. Maybe biotech is so boring. I know.
I think far from it, there's just a wonderful amalgamation of so many disciplines coming together. I think it's very educational. Thank you for sharing your experiences with us.
It's a pleasure.
I have one question regarding, I come from healthcare, and when you are creating synthetic batches, do you make sure they are balanced to prevalence, or just balance across all different variables?
No, both things. So let me go again to the slide. This one. So when we run at the end with the optimal values for the constants that we were using, we created these 40 synthetic batches. We checked that two things. One was that these synthetic batches were generated not from zero to the final process, to the batch time. So we created values for the synthetic variables, again, hour per hour, because here at the beginning, you can see that there is some dispersion at the beginning, and it seems that this model was good just in a very short time, in the middle or at the end, a little bit. So we used this synthetic values generated hour per hour, because when we use the same process for the full process, the data is not too good. So we used both things, the prevalence and also variable by variable that were under control for the reason, because we were able to check time windows per time windows, the validity of all the values produced in order to have consistent batches.
So do you think the results are biased with the current prevalence, and means that when you are using synthetic, right? So I just wanted to get an understanding of how using synthetic really helped you, or with the original 4 experimental batches, the results would have been the same, and the synthetic batches are biased towards the original experimental batches.
Good point. Well, we got some limits, as you can see in the dash lines. Yeah. These limits were the best batch that we had and the worst batch that we had. In order to define the two limits, and we try to create all these batches between these two limits. So maybe we included some bias, maybe, you're right, when we decide which were the right values of the constant that we were using for the scientific models, generation models, we decide the C1 is a constant of the model and C2 equals to 100, because the other ones that we used, and many other ones, were not responding well under this wrong and optimal batch. Yes, you're right. Probably we introduce some bias because we decide by a human perspective this value. Probably you're right.
Yeah.
Let me take some notes because I would like to discuss that with my colleagues. Thank you for
sure.
Thank you, Dr. Pawar. Any other questions from the group?
I've got one. Say, Tony, when you're describing the improvement of the process, the collection of blood plasma, and talking about, for example, the overall duration, and you opined, you complained about the lack of contextualization of individual data points, and there is a lot of implication there about what better contextualization could provide. If you didn't have time to go into that, but it seems to me that what you mean by the contextualization of off-process data in that case would be similar. Is it true then that the way you would handle this contextualization mathematically, that it's similar to adding more independent variables in a biological process control problem? That is to say, we have a measurement of glucose, and if it's not contextualized, there it sits. But if we say, well, we have a measurement of glucose, and at the same time we understand the alanine level, and we understand the pH, and we understand other independent variables at that time, would you consider that contextualization of an independent variable?
In some cases, yes. In other cases, not.
Oh,
for example, we didn't find the phase, for example, first centrifuge first filtering, second centrifuge, second filtering. Or, for example, what happens when there is a downtime because some filter has to be changed, has to be replaced. This context was not included in the data. But as you mentioned, we could infer because we realized that there were downtime too much long as usual. And we detected something thanks to the anomaly detection. But at the end, if the data was already contextualized, for example, because there is a maintenance operation there changing some filter, that could help a lot. So I think that, as you mentioned, Bill, I think that your point is very good. We can infer some reasoning from the real data. The problem or the thing is that if you have already contextualized the data, you can accelerate the process because you can realize of something that can help to you to find out some root causes, for example. But you're completely right. Sometimes the contextualization is not needed, but accelerates a lot the process. That I am answering your question, Bill?
No, but I'll ask it in another context.
Oh, sorry. I think that we have another question. It says, the agitator speed recommendation by random forest seemed a little noisy. Yes, you're right. It was really noisy. Also, what optimization algorithms were you using for making these recommendations? Let me go again. You're completely right. But you can see the black one is the agitation speed. And the blue one is the real one. What happens? In the real life, when you are changing the agitation speed, there is an inertia that is producing some hysteresis process where there is an inertia that you have to fight against. Well, the thing is that the prediction doesn't contemplate this inertia and is providing the optimal in every single moment, independently of the capacity of the agitation speed in order to achieve the expected speed. So for this reason, you see here that the usual one is constant because there is some effort, some resistance in the biomass to get this speed. But the algorithm doesn't know anything about that and is providing this, well, let me say noise, but this is not noisy, it's they calculated automatically depending on all the rest of variables. The second question is, what optimization algorithms were you using for making these recommendations? Mainly random forest and a variant of the random forest, including some exponential smooth processes, because there are many variables that they are working in an exponential way. But mainly was a random forest. And the third question is, how early does the anomaly detection algorithm detect anomalies? The answer is 10 minutes in advance.
Thank you. That seems very impressive. 10 minutes in advance.
Yes.
Thank you, Tony. So we're just kind of wrapping up. I had a couple of quick slides to go over with the group. Tony, if you don't mind. Thank you. So, so we have a couple of talks coming up for the rest of the year. On October 8th, we have a talk trying to figure out the final logistics. We'll let you know very soon. And December 3rd, we have Dr. Grossman from the FDA speaking to us about things like gold truth and gold standard and reference standard, those kinds of aspects for the evaluation of different algorithms. Next slide, please, Tony. Yeah, and just to let you all know, we are looking to your input on topics that are of interest to the group. So, as you know, and as Simon may have also mentioned, we have a survey going out pretty soon to all of you, soliciting topics as well as ideas that you think you want to hear about. So please do let us know about what you would like to talk about in this gathering. There are some other ways for us to engage in the community at large. One is, of course, the Xavier AI Summit that's coming up in November. So please keep an eye out for that. And as many of you are aware, we have multiple working teams within the Xavier AI group for AI in operations, GMLP, as well as AI at point of care, all of which is quite active in terms of community engagement, and lots of people are contributing to it. So if you're interested, please feel free to join us. Also, there are some very nice guidance pieces coming out from many members in the group on the Xavier AI blog, and the link for it is also mentioned here. So the multiple ways for you to be engaged, and really request all of your participation in this going forward. With that, I'll again close by thanking Toni on such a fantastic talk and educational perspective on two very interesting yet disparate problems, and how machine learning and AI has a role to play in CPV and those kinds of problems. So thank you again, Toni. Really appreciate your time today.
Thank you so much for your patience.
If there are any questions that the group wants to ask, maybe this is the time to probably unmute yourself, but if there isn't,